Central dogma. What is a gene? Well, in other terms, what is the information in the gene? Gene stores information, and that information can be transferred to offsprings. And that gene's information is actually the information of a protein. Well, protein's primary structure means the sequence of amino acids. That information is stored in the gene. Then, the original information of DNA that should be somehow transferred to protein, this relationship we call gene expression. The information of a gene is expressed in the form of a protein. In between DNA and protein, another molecule is involved. It's RNA. DNA contains many genes, and one gene's information is to make one protein. So maybe gene by gene, they may need to choose which gene will be expressed. So among many genes in DNA, only one gene's information is transferred to RNA. This step is called transcription. Transcribe means rewrite one information in another place. So that means from many genes information DNA, only one gene's information is rewritten on RNA. And that information is eventually translated into protein to make proteins. So here the relationship is RNA is a temporary information system of DNA. Now, to make DNA copies, so the DNA should be made by DNA polymerase enzyme, and then DNA polymerase utilize one strand of DNA as a template, and then make the other strand of DNA. That's why this process is DNA templated DNA synthesis. RNA synthesis is a transcription. Here the enzyme is RNA polymerase, another protein catalyst. And in this case, they utilize one strand of DNA as a template and the product is RNA. So this is DNA templated RNA synthesis step. How about the translation? By reading RNA's information, that information should be translated into protein, and the enzyme name here is ribosome. Here, very interesting thing is, ribosome is not protein. The major body of ribosome is composed of RNA, and this RNA is called as rRNA, ribosomal RNA. To compare to this, the rRNA, now this information RNA is called as mRNA, messenger RNA. It's very interesting to see that the important enzyme to make a protein, the ribosome, is not protein. It's RNA-based molecule. It's so interesting, isn't it? Now, here's the central dogma of biology. In biology, looks like DNA is original storage information, and this information is transcribed temporarily into mRNA, which is a more unstable molecule than DNA. And then utilizing this mRNA information, protein is synthesized. So this flow of information to make machinery systems. This is what we call central dogma of biology. If DNA is an information storage device, how much information can be stored in the DNA? To calculate this, let's think about the binary system first. Our computer system is a binary system utilizing 1 or 0 for the storage of information. If it is a 2-bit, it's a 10, 01, 00, 11. It's a 4 kind of information can be stored. If it's a 4-bit, it's a 16 information. 8-bit, that's a 256 information. If you have an n-bit, now the information, the number is a 2 to the n's. So to simplify, if you think about the 2 to the 10th, That gives us 1024, but that number is similar enough to 1000. 1000 is approximately 10 to the 3, right? So that's why to make the calculation simple, 2 to the 20th is easily you can convert approximately 10 to the 6, that means a million. This can help you to simplify your calculations. Now let's think about the minimum bit number for assigning 20 amino acids. Well, 4 bit you can get only 16, so at least 5 bit which can give you 32 different informations. Then maybe minimum you need to have a 5 bit to assign 2 amino acids, isn't it? Another thing that you need to think is in binary systems, if there's a all mixture of a 1 or a 0, then where is the starting point and the end point, right? So that's why to assign that, you need to assign at least one position, maybe same number. 0000 is kind of a starting point, and 1111 is maybe end point. Some information promise we need to have. Then out of 32, at least one sequence you assign for starting point, one sequence for ending point. Then out of a 5-bit system, you have a 30 information you can assign. In a similar manner, we can think DNA is quaternary system because it has A, T, C, G, 4 different information, the bit. That's why it has, comparing to the computer's binary system, DNA system is a quaternary system, we can think. And then DNA also has a direction. So the order of the reading frame is we read from 5' to 3' end. Now, if you think about the information size, 1 bit of DNA, it has a 4 information. Then 2 bit, 16, still that's not the high enough to cover all 20 amino acids. 3-bit, 64, now it's more than 20. The minimum bit for the 20 amino acids, yes, we can think that's at least maybe 3-bit of DNA may assign one amino acid uh, in translation. So that is true. And we found that 3 codon make one amino acid, and amino acid also has the direction, so we can assign from N terminal to C terminal. And that's why we can call DNA is like a 3-bit quaternary computer systems. Now, if you have a n and the n base, then the information number is 4 to the n. So that's a 2 to the 2n number of information we can store in the DNA. So 3 codon can assign one amino acid out of 64. Seems like one starting point is assigned here and 3 stop codon is assigned here. So the other 60s are assigned the irregularly to the amino acids. Usually simple amino acid has more chances of the having this codon, and rare or more expensive amino acids has been assigned lower chances. And these two scientists assigned those genetic code and 4 amino acid. In the beginning, they synthesized the UUUUU, and this is RNA molecules. And utilizing this RNA molecule, they found that this RNA can make the protein phenylalanine. So this is the first code they decode from the RNA to protein structures. And if you go to the NIH in US, there is their original lab notebook. In this lab notebook, they tried to assign which code assign which amino acids. They found one by one and figured out their full scale of the coding system, genetic coding system to make proteins. Of course, they got the Nobel Prize in 1968 for this achievement.